nanohub.org, online simulation, and more for nanotechnology.
Today's webinar will be Murnalini Pattarkine. She's a professor and chair of the Nanobiotechnology Initiative and director of the Capital Area Biotechnology Partnership at Harrisburg University of Science and Technology. And today she will be, was kind enough to share her time and talent with us for this webinar. So we're excited about nanotechnology workforce training on a shoestring budget. This webinar is hosted, you see the two logos up top there, by the NACK network. Basically, NACK is a NACK and also the MinTech National Center. We're both funded by the NSF Advanced Technological Education Directorate. We're Regional Center for Nanofabrication Manufacturing Education. NACK is at Penn State, and we're part of a larger organization called the Center for Nanotechnology Education and Utilization. We're hosted in the Department of Engineering Sciences and Mechanics. And the MinTech, or the Micro Nanotechnology Education Center, is again an NSF ATE center. It's the national center. It's hosted out of Pasadena City College, way across the country in Pasadena, California. So I am the host. My name is Bob Ehrman. I'm a consultant for the NACK Center at CNEU. Our co-host it will be Vishal Sirwade. He is an assistant teaching professor here at CNEU. Vishal, you just want to say hello to everyone. I know you just came off class.
Yep. Hi everyone. I'm excited for this talk, and thanks for hosting this, Bob.
Okay, no problem. And you can consider me as the play-by-play commentator, and Vishal is the guy who's the color commentator who kind of knows what's going on. He's the one who's going to be interacting mostly with Leena. We call Leena, or affectionately Leena. Or Dr. Pattarkine. And then we have Dr. Pattarkine, who is the professor and chair of biotechnology, as I said, at Harrisburg University of Science and Technology. And with that, I've known Leena for, I don't know, many years, and she's been a prime supporter of CNEU, and I'm really happy to have her here. So Leena, would you take over the reins and start the webinar?
Absolutely. Good afternoon, everybody. Thank you, Bob, for that kind introduction. And yes, it has been a nice, collaboration with the CNEU Center for past 14 years easily. And Bob and myself, we have done a lot of, series of workshops within Pennsylvania. So it's a good, very fruitful collaboration, and I'm happy to be a part of this webinar. So thank you for giving me this opportunity. And the way, as an overview, what I'm trying to say is nanotechnology, taking into the nanobiotechnology area, and then trying to figure out, what are the challenges, what are the concepts, what we need to teach, and creating a workforce that can be customized, for the industry where they are looking for those skill sets. And on the top of it, doing that on a shoestring budget. So that's the idea here, and I'm going to share whatever I have developed over the past several years. And the reason for me to be able to include or get engaged into workforce skill development is because of my grant that I direct, the Capital Area Biotechnology Partnerships. This is a consortium of academia, industry, and career technology education centers put together for creating a pathway that can train the students it was a 2+2+2 grant. So I have been working with the high school population, community colleges, 4-year degree program that we have in biotechnology, and now we have a master's in biotechnology. So we basically take care of or cater for the workforce skill development from high school all the way into the bachelor's, master's, and industry. So that has given me a lot of insight into my background is I'm a trained biochemist, and my PhD work was in protein immobilization and protein. Now I can call it as a nanobiotechnology. So that has given me some insight into what is required and how we can translate into modules for workforce training and how to customize it for a certain industry. So I'm going to share some of the stories with you here today. So can we have the next slide, Bob? So meaning when we say, typically the whole story is revolving around these three concepts, like what, why, and how. So what are the concepts that we need to cover? Why study in this field of nanobiotechnology? So where are the applications? Where are the career options? And how to do that? So how to create a trained workforce? And then, as I said, doing that with a shoestring budget so that it becomes affordable and manageable for any institution, irrespective of what kind of resources they may have. So it's a very affordable and approachable, cost-effective strategy that I have been able to design and develop, and has been a good feeling that it has been rewarded with students getting placed in good companies and getting jobs, and collaborations coming to us and sharing their knowledge and basically growing it from the sharing basis. So I'm going to share some of those things. So let us move to the next one. And this is something that probably everybody on this webinar already knows about it. The reason I wanted to use that is because this gives this lower, left corner, that is what is the Nanobiomaterials. And some of them, you can see that we have been able to conjugate or, connect with the nanomaterials, which are synthetic ones. So natural nanomaterials and synthetic nanomaterials, and how we can combine these two to create academic, resources so we can train students. So when we say nano, everybody knows nanotechnology, and what happened is for a long time, the nanotechnology was mostly catering for the semiconductor and material science industry. However, in the recent, few years, like maybe 15, 20 years, it has been realized with a lot of enthusiasm and embraced with a lot of enthusiasm that the same concepts, when applied to life science oriented projects or devices or technologies, that has a much wider application rather than just keeping it in the semiconductor and material science industry. So when we say nanobiotechnology, loosely, if you can say that whenever nanotechnology principles are applied to processes related to living systems, you're using something which is from the living system and creating a technology that can be applied for living systems. That is what is being covered here. So the very first thing is, whenever I hear that nanotechnology and nanobiotechnology, it is new. What is new? I'm going to share that in a couple of minutes. But when we say nanotechnology, nanobiotechnology essentially is not necessarily new, and I'm going to share a couple of examples so that I can validate my statement there. So when we say like an organ level, the human body, so I have picked up three basic, very important, meaning everything is important in the human body, but then I'm using these three examples. Heart. What is the function of heart? It's essentially pumping blood through the body for every peripheral, every single cell in the body gets the blood supply. It's critical because the blood is the one which carries the nutrients and oxygen and all the necessary supplements, and cleans up the body, also collects all the metabolic waste, carbon dioxide. So it's a kind of circulatory system part. So it's, in a way, we can consider that heart as a mechanical pump. It's pumping blood through the body. All right. Then look at the kidneys. What's the function of kidney? They essentially filter things out so that what your body can recover and conserve and reuse. And what needs to be thrown out, that is what the kidneys do. And lastly, the brain. It's a processing unit for the body. So receiving signals, receiving stimuli, interpreting that, and necessary actions, whatever our body can do. So movement, protection, defense, or any kind of action, or getting the sensory impulses, cognitive functions, all of those, in addition to controlling the different systems in the body, which are neural controls. So three major things. So kidneys, if we can call that as a filtration device, and brain as a processor. All right. So let us look at the thing that when we look at the artificial things, when we look at the pumps, mechanical pump, you can completely be surprised that your heart functions even before you're born, meaning even in the embryonic or gestational state, the heart is functioning. So unless there is some kind of major condition or situation or injury or accident, that mechanical pump functions to pump the blood in your body 24/7 as long as you live. So there is no other mechanical pump that can match that kind of superlative performance. When we look at the kidney function, it's a filtration device. So I'm using some picture, water pictures. So when we look at the water filtration unit, every so many gallons, you have to replace the cartridges so that you can have a functional, unit that can filter things out for you. Look at the kidneys. As long as you live, again, 24/7, they function in your body. And the brain, microprocessor. So if we have 20 or multiple functions on your computer, it might be a high processing computer and good quality computer, but even then you will see that things can slow down because you're running parallel functions, parallel things are being processed. Look at the brain. You are able to kind of do multiple things. Basically, you can be working you can be teaching in a classroom, or you can be sitting in a classroom learning lessons. You will be thinking about the homework. If you are working, you are thinking about the work related things, or something personal level. Nothing slows down. So my point here, what I'm trying to make it is, when we say that the nanobiotechnology is new, unfortunately, I don't agree with that concept. The human body is essentially made of nanoscale materials, and the function of those nanoscale materials is far, far superlative with respect to the consistency, reproducibility, reliability, efficiency. So when we look at these kind of things, so our bodies are essentially nano-enabled to begin with. They are inherently a complex of multiple nanoscale structures functioning in concert relentlessly throughout the lifetime. So when we look at the three major things that I'm going to focus on for the nanobiomaterial thing, first one is the DNA. If you take a double helical structure, it's a cylinder and 2 nanometer diameter. And we all know that the DNA is related to the hereditary function. So everything, a system or body, whether it is a bacteria, animal, plant, or human being, your DNA has the information as to what that system can do. That's the ability or the capacity. Proteins, 200 to 500 nanometer, size range. So they are still nanoscaled. These are actually the worker molecules. When we look at the body functions, whether I'm talking more specifically about human system today, but we can easily relate that for any biological system, right from bacteria, single celled, all the way to human, which are trillions of cells. So these are the worker molecules. They practically do everything in the body. And the other most important thing is the cell membranes. Whether you talk about the prokaryotic cells, eukaryotic cells, these membranes, these are the gatekeepers. They regulate what comes into the cell, what leaves the cell. There can be some kind of reliability. There can be some kind of baseline understanding that how the cells are supposed to function. So when we look at these three major biomolecules or three major, nanoscale structures, which are critical for any living system to function. So these are essentially at nanoscale. So I have had research experience for 20 years before I started teaching. And now when I look back, I can easily say that whatever research I have done essentially fell under or it is under the umbrella of nanotechnology or nanobiotechnology. So once I realized that, then I started thinking about it and I said, what do we do in nanobiotechnology? So practically we learn from the nature. So study those natural systems which have a kind of impeccable performance and impeccable control on the things, how they function. So we study them, extract that knowledge outside the system so that we can apply that. So when we say there is a cancer, so what has gone wrong, how you can treat it, how you can image it, how you can diagnose it. Once you understand the critical factors that lead to the condition, you can study and reverse engineer that to either create a new drug for treating it, a new diagnostic device. So the COVID thing, the vaccines are biotechnology products. RT-PCR that people did, that is a biotechnology technique. The rapid home-based antigen kits, that's a biomedical device that uses, antigens and antibodies and again, nanoscale structures. And once people realize this thing, that's the reason now there are so many industries which have already jumped on the bandwagon regarding applying those nanoscale principles or studying them so that you can create very innovative technologies and solutions. So currently we can see that being applied in health, medicine, biopharma, food and agriculture, environment, energy, water quality, plant, practically any aspect that impacts the human life. So that's the reason that there is so much of scope, so much of need for this kind, because industry has already recognized that. And the best solution is that our best advantage for using nanoscale material is that it has absolutely superlative sensitivity. It has absolutely superlative ability to make a resource conservation, as they call it. So instead of having to use multiple or volume wise or mass wise, you can use much less of the material and still get the same kind of output. Now, when we say, how to, then we have realized this thing. We know that this is the application and there is a scope and careers can be basically benefit from going in this direction. How do we convey? What is the skill? What are the skills? What are the concepts that we need to cover? And the fundamental difference is that when you look at nanoscale, properties change drastically. So when you look at the bulk material and the nanoscale material, they will have a completely differential, behavior. And that is something that needs to be identified, that, that nanoscale properties are different. And because they are different, they change. Obviously, you have to teach that in a different perspective and say that what are the ways, what are the techniques, what are the skills that enable us to study those nanoscale structures so that we can identify their functionality, we can use them and kind of use that in the process of creating innovative technologies. Now we have already seen that there is a concern about the nanomaterials which are synthetic ones like quantum dots or carbon nanotubes, because these are man-made, these are not naturally existing materials. So the concern is that when you use it and they get released in the environment, what is the impact? Are they going to be negatively impacting the ecosystem in human health as well as the environmental health? When we look at the nanomaterials, which are the nanobio, such as DNA and proteins, these are naturally existing materials. So when we look at those, they inherently fit that biocompatibility, space, that you don't have to worry about them thinking about these are artificial and they may have a negative impact or toxic, side effects. So how do we teach that? So the applications, you can see that I created these small sheets to just give a brief, overview as to, where is nanobiotechnology with respect to multiple industries or multiple fields. So you can use the nanotechnology materials, you can create conjugates, either use protein by itself, you can use DNA by itself. Sometimes they will have a conjugation of artificial, like gold nanoparticles with proteins, gold nanoparticles with DNA. And that has been very useful and very much revolutionary, if I can call that. So you can create nano sensors, which you are seeing that. And what people have done is they studied, say, like how the nose, the sense of smell functions in the body, reverse engineer that into creating applications, say, for a wine tasting. So it's a cartoon which is exemplified as a algorithm based electronic nodes or nano sensor nodes. Here, this is the packaging material for the food, so you can put nanomaterials or particles which are completely safe, let me say that. And they are basically embedded into the packaging material, and they are sensitive. So if there is a spoilage, if there is a contamination which is pathogenic bacteria or something is gone wrong with it, it will develop a color. So if the sheet of the packaging is completely clear, it's completely safe to consume. Any kind of color, that color coding can be different. We won't have a time to go into details of that, but that is clear indication that something has gone wrong with the meat or the packaged, food product, and it's not necessary to. Basically, you don't need to taste it, touch it, or feel it or smell it. It's a very clear, visible indication that something has gone wrong and that's not supposed to be consumed. The one in the center, you are looking at something which is called as a nano spray or nano laminate. So instead of using a saran wrap, the spray is essentially completely edible. You can spray it with the fruits and vegetables, mostly fruits, because it's slightly carbohydrate rich, sweeter tasting thing. But that can essentially preserve the food on the table without refrigeration. So these are some kind of mind-boggling applications and things like biosensors and carbon nanotube based biosensors. So those are the applications. Let us take a look at the medical field, where we have seen a lot of even commercialized application. So this is something that is a smart material, as we call it. It's a combination of like the way we have a quick fix. You can have a super glue and it will sense once it is injected at the site of fracture. And it has the ability to sense that it is within a biological system. It is responsive. It is able to sense the body fluid inside the body when you inject it at the site of fracture. It will harden, creating a cast, rather than the cast that you can apply from outside. For some complex fractures, it can work in a very effective way. The smart cloth, again, it's a bandage which carries some antibiotics and prime first aid, components. So only when it comes in contact with blood, it will release those, antibiotics and clotting things so that it can control the blood loss. If you dip it in water, nothing happens. So it has been designed and engineered. The nanoparticles, in addition to that, meaning people are familiar with the nanobots, meaning which is something that people have seen for a long time and heard about it for a long time. That's not something that is currently happening, but easily can happen. So ability to inspect the body and say what is wrong and intelligently deliver, whatever is the therapeutic medication or intervention. So these are completely, absolutely out of the world concepts, but they are very much reality for the medical field. Environment and energy, obviously you can see that, foldable electronics or backpacks which have the solar panels. So if you are hiking, it's a very popular, product in people who are hiking rigorously, seasoned hikers. Battery storage devices, biofuels, water filtration technologies, membranes used for water filtration. All of these are benefiting by using nanomaterials or nanotechnology. And even for a common man, you can easily see that these are a day-to-day products. So the water-resistant fabric, whether you want to use it for the attire or it can be a furniture protection, doesn't matter. That is very popular. The dental things, when you look at the fillings, the kind of precision and kind of materials that are used, these are biocompatible, they blend with the skin. Makeup, all those, advanced stage serum containing, which are age-defying serum or lasting makeups and things like that, or hair colors. All of them, they use nanomaterials. All right, so we have seen that, there is so much of applications which are already out there, and there are more that are coming out. So what is typically being targeted? So as I said, proteins, the DNA, and then conjugates of nanoparticles, such as, gold nanoparticle, silver nanoparticle, quantum dots, either with proteins or DNA. So typically these are the majority of the things which are being exploited seriously and routinely for the nanobiotechnological application. So it may be cancer diagnostic, cancer imaging, targeted drug delivery, controlled release mechanisms, gene therapy and cell therapy. All of these, they are essentially under that umbrella, DNA, protein, and conjugates. So I'm going to spend a few minutes highlighting protein-based nanobiotechnologies, and the reason is that there are certain devices, which have used the nanomaterial for the diagnostic purpose. So if you can see, there is a list of all the functions that proteins perform in a body. In a body meaning human system. So they support the body in the sense like whether at cellular level, there are proteins which help the cells maintain their structure or morphology or shape. And also when we look at the body as such, the musculoskeletal system, that is. So the bones are essentially supported with muscles. And muscles are nothing but, proteins. Movement, again the same thing, muscular movement, the contraction, and you will see that example coming up. Transport, getting things in and getting things out of the cellular system. Maintaining the buffer pH thing so that the body can function in an optimal way. Hormones like insulin and other kind of hormones which regulate the body functions. These are proteins. And of course, meaning the antibodies are the defense mechanism, they are again protein in nature. And because the proteins are so diversely applied, or they do so much function within the body, or body depends on proteins. All the enzymes in the body are proteinaceous in nature. So here are some natural protein-based structures which are very critical for the functioning of the cellular structures. So if you remember, I said that the transport or what comes in and what leaves the cellular system. That exchange happens through certain mechanism, and many times those proteins or the channels or the transport mechanism, they are proteinaceous in nature, and they function by small manipulations. Either it can be an ion gradient or a voltage gradient. So if you can see, and very simple, meaning we have to understand that nature is by far the best craftsman. The kind of craftsmanship and the intelligence that is packed in the natural system is beyond comprehension. So all it does is, by changing the small voltages, and these are very small quantities. These are tiny structures, but they are efficient enough and they are sensitive enough. So by changing the potential across that, membrane, it can either open up the thing so it can accept and bring things inside, or it can close the thing so that it will not let things either leave or come. So that is some mechanism that controls, the body makes sure that it doesn't get too many things or it doesn't lose too many things. So it's a regulated thing. The exchange happens in a very regulated way. Then I was telling about the muscle contraction. So there are two basic proteins in the muscular movement, and they are called as actin and myosin. And here is the thing, that this is the resting muscle, and this is a contracted or muscle. So what it happens is things just, they slide past each other. So when they are relaxed, they are like this. When they are contracting, they slide between each other. Very simple mechanism, but absolutely stunning example of how that protein mechanism can help you just to move. Even when you blink, it's a muscle movement. And what we have seen is that these are very fast. When you can see that the muscles contract 20%, that means 1/5 of the length over a period of, few milliseconds. And we don't realize, we don't pay attention to these things which are functioning in the body. Because as long as things go well, we don't really realize it. But once you get some kind of sprain or there is a muscle, which is tore or something like that, then it will painfully make us aware of, how superfine way or how smoothly these functions within the body. And this is something that is completely basic of, how the body generates energy. It's an enzyme called as ATP synthase. And I won't get into details of that, but this is like a reversible motor. So it turns clockwise, anti-clockwise, the units. Like there are, if you can consider this. So there is a proton gradient, and protons is what basically hydrogen ion. So it's an electrical gradient across the membrane. And that is what is utilized for creating or synthesizing ATP, the energy currency for any living system. So this is the example as to what is happening is simple thing. Again, open close we saw for the exchange mechanism. Here it is for the energy synthesis. It is moving in a certain way. It's like a gear. It moves in a one way, clockwise, anti-clockwise. It will either make ATP or it will synthesize the hydrolyze the ATP so that it can be used for driving body function. Just imagine how intelligently nature has designed those systems. So these are some of the examples, and there are many more I can quote, but we are limited on time. So I'm just going to give a couple of examples there. So where have people utilized, where has the industry utilized these nanomaterials for commercialized technologies? So obviously biosensors, when we look at the glucose monitoring device or a pregnancy testing kit. These are the biosensors. We choose protein, and they use immobilized protein so that the protein won't get washed away or it will not move. It will be there when we want to use it. Then there are DNA protein conjugates used for diagnostic purpose, medical imaging, drug discovery, drug delivery. And then on the top of it, we can functionalize the nanomaterials, conjugate them with protein so that you can have a targeted delivery mechanism or targeted, imaging purpose or diagnostic thing. So these are some of the very key kind of examples of where proteins as nanobiomaterials are being used very successfully, very effectively, very sensitively. So pregnancy testing kit, it's an optical biosensor. It will it will design or give a output based on the binding thing. And I won't go into detail of how that functions, but this is simple, like one line or two lines. That's the idea that can detect, if the lady is pregnant or if she's not pregnant. And of course, this is a primary screening. That doesn't mean that, you don't really need to go to a physician and, get the consultation done. But this is a very quick kind of, at-home test. You don't need to go to a diagnostic lab. It's very convenient, very affordable, and relatively quite accurate. The second example, and I'm very fond of this example of glucose monitoring device, the glucometer that is used by diabetic patients to test the blood glucose levels. It's again a point of care or at-home diagnostic device. And not only it is a biosensor, but it's a great example, the strips that are used typically for testing the blood glucose. That strip itself is a great example of nanofabrication. I think there are 6 to 7 layers in that small strip, which is no thicker than, your probably photo paper. So within that small thing, which is not a few micrometers, there are 7 different layers of, materials that are fabricated. And then it also involves protein immobilization. So it's a great example of, it's a biosensor, it's an electrochemical biosensor, at-home device, nanofabrication example, and protein immobilization. So many concepts can be basically covered with that. And it's a cheaper device. If you want to buy it for the lab purpose, it's quite affordable. So we have seen the ability of these nanomaterials to be used or to be researched and to be applied as a nanobiotechnology material to create devices, technologies, whatever it can be. Obviously, that is something that is very promising. So when we look at the skills, where do we start teaching it? So obviously, as I said, the students, we need to teach them the concepts. What are the concepts, the unique properties that are exhibited by the nanoscale materials? And how do they connect with the biological system? So when we look at these things, once we create that path and then expose the students to those concepts, it's easy for them to understand that how nanobiotechnology can be a career option, can be a path for them to learn or education and get the education and get into the industry. So when we see that, we need to teach them the skills that will enable them studying the properties and explore the applications of the nanomaterials such as DNA, proteins, membranes, cells, and then thinking outside the box, because nanotechnology definitely is the next industrial revolution. So making sure that they get the basics, the concepts, understand the skills that are typically used by the industry. And the industry may be very precise and very high throughput and very automated and all that, advanced kind of thing. But when you teach them in the classroom, getting them the concepts and letting them understand that, okay, if I'm doing this, this is how it's getting translated into the commercial application by the industry. That is all we need to focus on. So when we look at the things, protein immobilization is one of the big techniques that is used. Protein characterization, protein isolation, and estimation. So when you say that it is there, how do you identify how much is there? How do you know that it is still functional even when you have immobilized it? And how do you know that the system is functioning properly? Same way, when we look at the DNA-based technology, and in most cases, you will see that the nanoparticle and DNA conjugate is what is being used. And the most popular nanoparticle in this application seems to be gold nanoparticles. So teaching them what are the nanoparticles, how to synthesize them, how to characterize them with respect to their size, their optical properties, how to create the conjugates. That means how you combine them with proteins or DNA. Once you have done that, how do you characterize that and make sure that it's not just assumption, but it is actually conjugated and it is stable and you can use it. So that surface functionalization or changing the surface property so that you can customize it for a certain application. That is a big technique. And in most cases, as I'm saying that I'm focusing on cost-effective strategy. So believe it or not, but the analytical techniques of absorption and emission spectroscopy comes very handy because there are many optical biosensors and they have been very popular and easy to kind of conceptualize the ideas and teach it to the students. Let them work on it. So it's very affordable strategy. Now, when we say that, these are the things we can do that. The challenges are three major challenges that I can say that I have realized over the past 15 years that I'm doing this. Very first thing is the lack of awareness, whether it is nanotechnology or nanobiotechnology. In the K through 12 space, there is no strategic standard that says, nanotechnology or nanobiotechnology. At least in a couple of states have actually included that as a standard, but many of them, they don't. So there is a lot of awareness that we need to start from there, actually telling them what is it and why you should study it. On the top of it, the second thing is that there are misconceptions. Because when we say that, nanotechnology and multi-million-dollar industry setups, people tend to think that this is out of reach for a common, high school or a small university where you don't have those resources. So that is a misconception or that's a myth. And the third one is the access to those kind of facilities, meaning I have been working collaboratively with the CNEO, absolutely stunning, NSF center facility. If you want to take students, I used to take them there for the free trips and we work very collaboratively. But then if you're not able to travel, if you're not able to take the students to those facilities, what do you do? So that geographic distances and the barrier, that can be a barrier to letting them see how things happen and what is the thing. And the worst of the things is that fact that nanoscale materials are very small and you really cannot see them even with a high resolution light microscope. You really need very high-end instrumentation on an average. So when we say these things, so you cannot see that, but on the other end, I'm talking about, creating diagnostic abilities, which are, I can say that these are sensitive and you can count on them and you can trust them. So for the students, it's very difficult to comprehend that what you cannot see, how can you rely upon it? How does that happen? Then another example I will give you for the carbon nanotube. These are very lightweight, and then we can say they're flexible, but they are 50 times stronger than the steel. Meaning these are some kind of oxymoron situations that you say one thing and it is something completely different when you talk about property. So these things are some things we need to eliminate, break those barriers, bring them down so that students can actually start seeing things and start believing in the field of science. So using my research experience and the industry feedback and innovative thinking, we ended up creating certain solutions so that we can create awareness, let the students know about it, bring them into the area and let them be familiarized with it. So if they like it, they're going to follow that as an education path. So we have designed, the very first thing I did was a summer course, just 1 week long high school summer course, which will give them a taste of, like a taster thing. What is nanobiotechnology? What are the applications and where are the career paths? It has been very popular. It's a 1 college credit summer course for high school students. Then strategically knowing that there will be need for, higher level educational offerings, we created a concentration which is dedicated to nanobiotechnology, and in collaboration with the Penn State, CNU facility, we also offer a nanobiotechnology nano fabrication. So they do 1 semester at the Penn State, CNU as a part of their biotech program. So that is the educational one. Then design online resources that can introduce them to the skills concepts, and the very one, the one which is, since we talked about proteins, that is something that they need to estimate and quantitatively know. So that is one called as a Bradford assay. It's a protein estimation method. And I generated an online tutorial. And similar tutorials were generated for whole nanoparticles, like how do you synthesize, what are the structural characteristics and what are the applications. Biomarkers tutorial, because biomarkers are the proteins which can be specific for a condition like a cancer biomarker. Or for COVID, we had the spike protein from the virus as a biomarker. So based on these things, industry can create diagnostic technologies, imaging technologies. They are also used for the drug discovery and delivery mechanism, so multiple applications of biomarkers. And as proteins, they are nanoscale structures, so tutorials on what are biomarkers, what are biosensors, multiple of those resources. And thankfully, we have a terrific, technology support at the university, so we collaboratively work and generate those resources. So I'm going to quickly show, I think, we are almost towards the end of it, but I'm going to share this resource. Particularly, I have benefited significantly by using the tutorial, so it will walk the students through the basic things, what is it, and what are the components, what's the protocol, and we can kind of, move the students through the. So there will be an introduction, what is this assay, what are the materials, the key concepts, how do you prepare the sample, how do you document the things. So you can please show that. I'm So it will give a brief theory part. It will talk about the sample preparation, how you run the assay, what are the critical things, and the biggest part where students really find it confusing is the calculation, the math part of it. So I created a specific, Excel sheet, and it will walk them through, like once they report the thing, do the assay, get the observations, how to feed it into the system, and how to use for calculating the protein concentrations. So this is a complete package in itself, and there are additional things where they can see troubleshooting concepts and validation, statistical analysis. So it's a STEM kind of package that will give the science, the technology, the mathematics, everything is embedded into the thing. So just as an example of what I say, that we created those resources, and there are multiple resources like that. These are online resources, so people can always approach us and then we will be happy to share it with them. But this has been very helpful. So similar resources have been designed for biomarkers, biosensors, regenerative medicine, artificial skin, and gold nanoparticles I already mentioned. Then on the top of it, what we did is just letting the students know about it doesn't really help as much, because if it is a new field, not relatively known field, the parents need to be convinced as well. So we have done activities, with the students and invited parents, and believe it or not, sometimes the parents were more excited than the students doing the activities. Then simple card games or board games, which they can do as a literally a time pass activity. I designed that. But it has been so popular that my students, they will come back and ask me, can we take it over the weekend? We want to play it in the dorm. Meaning that's the kind, you need to get them hooked up onto the concepts. And then because we have a bachelor's program, we have design projects which are utilizing these concepts like proteins and nanoparticles and diagnostic assays and biosensor assays. And these are conceptual designs, meaning they don't really have a high-end kind of fabrication aspect of it. But even when the students work on it, and they have been very cutting edge, like Alzheimer's, there is no diagnostic kind of ability right now. Preeclampsia, another condition where there is a fatality associated with it, the pregnant women who suffer from preeclampsia. There can be death sometimes if it is not monitored and diagnosed. There is nothing that is a quick and sensitive assay. So one of my students is actually working on preeclampsia diagnostic as a prototype. Pesticide detection in the wash water for produce, agricultural produce. So the farmers need something like that. So we have designed that. And also the bioprinting and the artificial skin and regenerative medicine. And we have been, the university has supported with some seed funding. So you can explore new ideas, and if it is something that is going to work out, possibly you can take it out and see if you can collaborate with the industry. So collaboration is a big, resource when you look at on a shoestring budget. And as I said, some of the things that I have routinely used are the gold nanoparticles, because it's a great example again of multiple concepts. So size dependent, properties, optical properties, and how that can be utilized in biosensor designing, conceptual assay development. I have used that. Applications, people already know that. What are the applications? Similar thing, very popular is the quantum dots. And the optical sensor, meaning UV visible spectroscopy and emission spectroscopy for quantum dots, and conjugating them and generating, because there is practically no lab that deals with biological labeling, whether it is for cancer or drug discovery, drug delivery, study of diseases, study of conditions, metabolic disorder. Quantum dot labeling has been very routinely used. So that's another one which we very significantly focus on. And then there are some funny things, which are fun-free things, not funny, which is the non-Newtonian fluids. That's a concept, again, for the nanoscale materials. And the best example that we have used is for the liquid body armor. And what I have done is, I'm going to quickly share this video where we actually created a big tub, filled with cornstarch.
Yeah, now we can.
So I technically filled the complete tub with the cornstarch non-Newtonian system. And the kids had a fun. So basically, these have been kind of innovative ideas. So when they saw that you can actually walk on that tub filled with the cornstarch, it's a liquid. So the concept of how it can be used for a body armor as a liquid thing. So what is the non-Newtonian fluid? How it responds to an impact or pressure application or force, and how that can be exploited for creating lightweight body armors. So just being kind of innovative is the idea here. So this is all with the capital area biotechnology partnership, which is the workforce training entity that we have, and these are some of the pictures from how we have engaged the students over the past 14-15 years, and you can see it's not only students but parents, and everybody is quite engaged, and it has been a lot of things. Lot of low budget activities that have led to a lot of things, and there you go. So I'm going to give it back to Vishal now.
So there is a question here: at what level of education the nanobio should be introduced?
Okay, so the question is very effective and good question, because when we talk about high school, it is a bit too late. So you should introduce them at the middle school level and let them see very simple fun-filled activities, but they can remember something about nanostructures and nanotechnology. Even if they remember these two concepts when they get into high school, that's good enough. High school, we have definitely much more ability for them to understand and work on projects, and the summer course has been quite popular, and we have had some students who have utilize the credit to come into my program as a bachelor student. It has also been accepted by other universities when they requested transcript. The students who went there, they got a credit for their bachelor's education. So those are the things. So you can, elementary school is a bit, it becomes too diluted, but you can still engage them, but middle school and high school definitely is.
There's one more question from an attendee, Richard. What are the names of occupations that one could expect to enter with the bio-nanotechnology education, and what are their pay ranges?
Pay ranges, that's a great information and great question. And the careers can be, typically when we say nanobiotechnology, there is no nanobiotechnology scientist. No, it doesn't really go with that kind of titles. It will be a scientist or engineer or R&D kind of person, but then within the job description, they can see that, ability to work with, nanomaterials or proteins and designing assays and those kind of things. So you can be a biomedical engineer, biomedical device engineer, you can be a drug design person, you can be a material scientist, you can be working with the instrumentation industry, because when we work on the materials, we need instruments, that can help you characterize things. So they can go into that. They can even work in a food and agricultural industry because the sensory devices that are used. You can go into environmental application. You can go into energy sector, water quality monitoring. So for biopharmaceuticals or drug designing, drug discovery, drug targeting, drug delivery, all of these are, multiple areas where we can. So basically biochemical engineering, biomedical engineering, bioinstrumentation, biotechnology, pharmaceutical industry, generic, contract research organizations where they produce things, device industries. All of those basically need people who have the expertise in using biological material. They may be a part of a team. You don't do things by yourself, but that is where the applications are. And the pay scale depends from the starting salary for a person who has a nanobiotechnology degree, bachelor's or master's, definitely it will change. It can be anywhere between 60 onwards, minimum. And depending on where you reside, if it is cost of living is high, it can change, but that is typically how you can start from 60 to 65 onwards.
Fantastic. Leena, I want to thank you once again. I really appreciate your collaboration and today's work on the webinar and all the work you've been doing at Harrisburg University. And thank you for being willing to share all the things that you've created and all the resources that you found with our audience. Really appreciate it. It's an excellent topic, and thank you so much for all your work. We really appreciate it.
Not a problem. I appreciate Mintech and you giving this opportunity to share, what we have. So in my opinion, if you share, you grow when you share. So that's the
Absolutely. Thank you so much.